Evidence-based medicine is built on a simple but demanding principle. The confidence we place in any treatment should be proportional to the quality and quantity of evidence supporting it. By that standard, the current market positioning of NMen as the sophisticated, modern alternative to niacin reflects an inversion of what the evidence actually justifies. The gap between what we know about these two compounds is not a subtle difference in methodological detail. It is vast. Niacin has been studied in humans since the 1930s. The clinical research base spans thousands of trials, including large-scale randomized controlled trials involving tens of thousands of participants across decades. We have data on niacin's cardiovascular effects, its safety profile at various doses, its long-term tolerability, its interactions with other medications, its dose-response relationships, and its efficacy across diverse populations and health conditions. This is the gold standard of evidence depth, the kind that takes generations of researchers to build. NMen entered the human research literature in earnest around 2018 to 2020. As of now, perhaps 10 to 15 small human trials have been completed, most involving fewer than 50 participants, most running for 8 to 12 weeks. And most assessing surrogate endpoints like blood NAD+ levels rather than clinical outcomes like disease incidence or mortality. These are pilot studies. They establish biological plausibility and safety signals. They do not establish clinical efficacy. In the hierarchy of evidence, they sit several rungs below what niacin has accumulated. Calling this a fair scientific comparison would be misleading. This evidence asymmetry doesn't mean NMen will never prove its worth. It means that current enthusiasm for NMen is running far ahead of current evidence. The longevity research community is excited about the underlying biology, and that excitement is scientifically legitimate. The mechanism of ND+ decline with age, sirtuin dysfunction, and mitochondrial deterioration is a compelling framework for aging interventions. But excitement about a mechanism is not proof of an intervention's efficacy. Consumers who want to evaluate supplement claims critically need a few basic tools. Understanding study design matters. A randomized controlled trial in humans is fundamentally different from a mouse study showing life extension in rodents fed experimental compounds. Identifying conflicts of interest matters. Research funded by NMen manufacturers and published in contexts where the funder has input deserves additional scrutiny. Recognizing the difference between a surrogate marker and a clinical outcome matters. Showing that a compound raises ND+ in blood cells tells us something, but it doesn't tell us whether it prevents disease or extends healthy life in humans. Niacin, with 80 years of data, is not a perfect compound. But it is a known compound with known benefits, known risks, known doses, and known interactions. For the vast majority of people considering ND+ support, starting with the compound that has earned its evidence base through decades of research is the more defensible choice. Adding NMen to that foundation if and when robust human evidence emerges is a reasonable upgrade path. Skipping the foundation entirely in favor of early-stage hype is not evidence-based, it's marketing-based.